You already know Adriana, but I'm going to make the introduction again. So it's my pleasure to have here Dr. Adriana Sanz Garcia. So currently Adriana is a postdoc researcher at Joshua Zweig's lab in Georgia Tech. And she joined the group in the very beginning of the pandemic, coming directly from Madrid, where she earned a PhD from the Centro Nacional de Biotecnología, a Spanish national center, which is focused on multidisciplinary research. There she worked with Susana Manrubia, and from the Universidad Autónoma de Madrid, Adriana got the master's degree in biophysics, and prior to that, she got a bachelor degree in biochemistry, biophysics, and molecular biology from the Universidad Autónoma de Madrid. Adriana's research line is focused in models made from very basic elements, and to understand the extension of these models to what we observe in nature. In summary, I would like to say her research line is very related to the main fields of this course, as you already know, and that's not by chance that Adriana is here. So please help me to welcome Adriana Sanz Garcia.
Thank you. I think I have kind of like, I see a delay in well, I think now I'm fine. So I am very happy to see you all again. Most of you, almost all of you. So I hope you are having like a really nice course. And well, I did this presentation in very fast. So because we are very busy right now, we are actually giving the same course, but the extended course of quantitative bioscience at Georgia Tech. So I have classes, etc., my research, and I just see here that I have like a mistake. So probably this presentation is going to be full of mistakes. So sorry in advance. So today I'm going to talk about what parasites do in ecological coexistence. So how they help to promote ecological. We are going to talk about viruses, and we are going to talk about satellites that are parasites of viruses. So this is my outline. I think it's a very ambitious outline, but I want to give you like an idea of what is microbial coexistence and how viruses help in microbial coexistence in different ecological niches. And the second part is more specific about project that I just recently published on satellites, how they can enhance viral coexistence. So first things first, let's talk about microbial coexistence. So here we have two microbes, two Paramecium, Paramecium caudatum and Paramecium aurelia. And Gauss wanted to study them and see how they growth in experimental conditions. So this is how they grow individually. So here we have the Paramecium aurelia that grows a little bit exponentially at the beginning, and then it reaches to a saturation. If you remember from class, this corresponds to kind of like a logistic growth. The same happens with Paramecium caudatum, right? So what they observe is that if they put together the two Parameciums, what they observe is the Paramecium aurelia wins and the other one kind of gets extinct over time. And this study was remaining kind of like a mystery for several years until Hardin put some kind of equations to this growth of these two Paramecium, and he established or summarized what is happening here in the competitive exclusion principle. So two species that are feeding on the same resource cannot stably coexist in the long run. So basically, the Paramecium that take advantages of the resources more efficiently is the one that survives and the other dies out. If we translate this into virus, if we extend this to microbes and virus coexistence, we have to talk about the Lotka-Volterra dynamics. So this was originally presented by Lotka-Volterra, is the predator-prey model. So we have two types of variables here, the prey that grows exponentially, right? And then it's eaten by the predator. So, and the predator, if they don't have the source or they don't have prey, they die out. So then you can see these oscillatory dynamics in which the prey starts growing in the absence of the predator, but then the predator starts growing. So then the prey decays. And then if the prey decays, the predator decays. So this is like characteristic dynamics are observed in many levels of ecological systems. So there is only two possibilities, one coexistence possibility, that is this center. So again, what I said, if preys increase, then the predators increase, and then if predators increase, then the preys decrease, and then you have these centers, stable centers, so the dynamics are circulating in this counterclockwise manner, right? So apply these to viruses and bacteria, we can propose similar models to Lotka-Volterra. In this case, we are going to propose a compartmental model in which we have three possible compartments: susceptible bacteria, N, that grow until the resources are depleted, that is like given by the K, the carrying capacity. And then these susceptible hosts or bacteria can be infected by a virus, right, a phage. So we have infected bacteria, we call it I. And then eventually these viruses inside of the bacteria kill the bacteria. So this process is called lysis. And with this killing of the bacteria, a number of new viruses emerge. We characterize this by the burst size or beta. And then this repeats over and over again, right? So if we observe to the null lines of the trajectories of the virus and host, again, we can see the Lotka-Volterra dynamics. So the host, the bacteria increase, then the virus increases, then you again see these kind of trajectories in which you end up in a stable center or an oscillatory dynamic. So much later, like a century later, more or less, Lenski and Bohannan perform these experiments. So they use E. coli, the bacteria E. coli, and T4 phage, a virus, and they put them to coexist together, and they could observe that, again, these oscillatory dynamics are present over time. So it was the first time that in this control experiment was observed these oscillatory dynamics. But then if you continue having these oscillatory dynamics, suddenly something happens. So they incubate this virus and the host for about 200 hours. So this, if you remember, this is many generations, right, of bacteria and many cycles of infection. And what they observe is that suddenly the host stops oscillating and the virus continues oscillating. Very weird. So what was happening here? What is happening is that this host corresponds to a resistant host that is in much higher density than susceptible hosts that are still like a subpopulation of susceptible hosts that keep oscillating with the virus. So this is what is happening. We are having coexistence also in not only in the short term, but in the long term, we have to incorporate the coevolution of the virus and the bacteria, and how this coevolution is happening or how it can be. So we can define this virus-bacteria coevolution as a interaction network. So at time T1, the initial time, we have one type of viruses and one type of bacteria. These viruses infect the bacteria, so the bacteria eventually evolves to a resistant bacteria. So these resistant bacteria cannot be infected by the original phage. But eventually, over time, this original phage also mutates. So it creates a subpopulation of phage that can, and so on and so forth. And in this case, what I'm doing here is that I am assuming that the new subpopulation of phages not only can infect the new resistant bacteria, but also the ancestral ones. So in this case, if we continue doing this over time, over the coevolution time, we end up having a nested network, a nested coevolutionary network, in which the original bacteria is going to be infected by all the emerging phages over time, and the new resistant bacteria are going to also increase in resistance, so less viruses are going to be able to infect it. So we can represent this interaction network as a matrix in which you have bacteria and phages. So in this case, this bacteria is infected by all the phages. We will call this like the original strain. And then here we have the original bacteria, the final bacteria, or the bacteria that have evolved resistance and is only infected by one of the evolved phages. But these networks or interaction networks, evolutionary interaction networks, can be different. In this case, I just painted here an example in which, again, you start with a pair of bacteria and phage. So this phage can infect this bacteria. Eventually, this bacteria evolves resistance. And then, again, the viruses can evolve to higher infectivity or another strain of the virus that eventually can infect this resistant bacteria, but it cannot infect the previous one. So kind of like you lose the memory of infection, let's say. So we can have these one-to-one networks or more like modular networks. So in here, evolved bacteria kind of like interact with evolved viruses in a very short term. So we can wonder, okay, how this coexistence can happen in these types or in different types of networks of interaction networks. So here I'm going to talk about a generalized model of virus bacteria coexistence. So again, we have here for bacteria, so we have the original one that is 1 and the original one is n and the final one is 1, and the phage. So we can define these interaction networks by the more generalist phage. So the phage that can infect all the bacteria, we can call it the phage which has the higher host range. And then we can also define the most resistant bacteria, that is the one that is only infected by one of the phages in the population. So we can express the dynamics that are going to be, again, very similar to the SIV model that I presented before, but now we have to include the interaction networks, right? The interaction network. And basically what we observe is, again, kind of like an exclusion principle. Exclusion principle, again. So we have two conditions for coexistence. One condition is that the bacteria that is more resistant has to have the smallest exponential growth rate. And on the contrary, the phage that can infect more bacteria, it has to have very specialized bacteria. So in this case, it will have like a smaller infection rate for the whole bacteria. So we can say that in the case of this phage over here, the phage one only infects this bacteria, right? And is very specialized on this bacteria, so it can allow to have a very high infection rate or infection ability for this bacteria. But on the contrary, this phage that can infect all the bacteria, like we can say that the infection ability for all the bacteria cannot be like so high. So we can have this like cascade of host range and host bacteria. And this is basically like a very strict condition for coexistence, let's say. So this is what we observe in nature. So here we have, again, an interaction network between marine and bacteria phages. So these are many species of Vibrio. Vibrio is a bacteria, and then different phages that have been isolated. And in this initial paper, what they observe is that these modules that you can see here in different colors happen in either because of geographic of taxonomical scale. So this means that probably this module that is here corresponds to one family of Vibrio, and then the phages that can infect this particular family of Vibrio. Another taxonomical level will be another family of Vibrio can be infected by this specific this specific phages, and so on and so forth. And these can be taxonomical scales or either geographic scales. So these Vibrio that live in, for example, in the deep, in a certain depth, or in the soil of the sea, they can be infected by this kind of phages. But the ones that live in the surface, they are infected by other kinds of phages, right? And then we can talk about the nestedness of these networks when there is overlapping killing. So this is like the same as we talked before for the host range. So if our phages have a higher host range, so they can kill many types of bacteria, this is going to be a byproduct of recombination. So this is how they, like, this is now this is the state of the art now. Like, we can observe different interaction networks for virus and bacteria, but they have these kind of rules on how they structure themselves. So I'm going to talk here about a small teaser of my work. That I'm not going to like elaborate on this, but I wanted to just put here a glimpse of what I'm doing right now. So we are working with real data in collaboration of Justin Meyer. So here we have, again, interaction network between E. coli, so bacteria, and phage lambda. And this is an interaction network over time. So we start with one strain of E. coli and one strain of lambda phage, and they evolved these two strains over time and see what your existence matrix or the infection matrix. So then, for example, at day 8, you have that, I don't know if, yes. So I don't know if this is incorrect. Sorry, I'm thinking about this. Yes. So this means that at day 8, viruses from day 15 can infect this bacteria at day 8. But the phages that are evolved more time for 28 days, they have a higher infection rate for this E. coli. So the colors here is the phenotype of infection. More red means higher infection or higher efficiency of infection. So you can see here, for example, in this particular case, that the phages that have been evolving for 28 days have a higher infection efficiency for very past phage. So it's kind of like a kind of a nested interaction matrices, like we saw in the other examples. And what we were wondering is how these interaction networks appear, and more specifically, which are the leading mutations that are actually letting these co-evolving coexistence networks happen. So for that, we normally, or what I am doing, is creating machine learning algorithms that can reconstruct these fake bacteria infection matrices. And from these models, I can forecast important mutations for the fake and the bacteria that in red are negative, or in blue, in green are positive for the model. So let's say that the green mutations in bacteria and the green mutations in fake, both together are, let's say, positive for reconstructing the infection matrices, and the red mutations in both the bacteria and the fake are the ones that are kind of have a negative coefficient for reconstructing the fake infection matrices. But I'm going to talk about, I don't want to go in detail about that. This was just like a little teaser. If you guys are interested on these kind of coexistence models on the coevolutionary pathways, you can ask me later, or you can send me an email. And today, what I'm going to talk, or what I want to talk, is about another project that I did during my thesis that is published now, so it's kind of like a long time. It's about how satellites affect viral coexistence. So in this case, instead of virus microbe coexistence, I am looking for how two viruses can coexist in one host. And in this case, I'm going to specialize in one particular host, that is plants. But I'm going to talk more in detail now. So first of all, let's classify defective subviral forms. So here we have a table of life-dependent organisms. So we have viruses that we just talked about. So these viruses depend on living system, whatever it is, a host. And then we have virus-dependent entities that we call satellites. So satellites are subviral entities that depend on a helper virus in order to replicate. By their own, they cannot replicate. An extreme case are defective interferon particles. So I don't know if you are acquainted of that, but defective interferon particles are just errors in the replication of a virus that encapsulate and then propagate. But because they are just like a fail in replication of the genome of the virus, they cannot infect by their own. They don't have all the genetic material necessary for having a successful infection. So they need from the helper virus or the original virus in order to prosper. And in between, again, we have the different types of satellite virus. And the interesting thing is that most of these satellites are emerging in plants. So plants is like a very, like, this would be our ecological environments, plants, from now on. So we can define the spectrum of the strength of satellite and virus associations in this little graph that I have here. So, as I said, defective interferon particles are just defective particles that appear in in viral infections, and they are errors in the replication of the virus, and they normally attenuate the infection. So the phenotype that you have as a result of having a population of viruses in which you have defective interferon particles is that the ability of the virus to infect is attenuated because these particles are kind of interfering in the infection process. But this interaction between the defective interfering particles and the original virus is very transient. So if you have kind of like a small bottleneck, you will basically all these defective interfering particles will die out. And in the other extreme, we have multipartite species. So these kind of viruses, multipartite viruses, are viruses in which their genomes are split into pieces. Contrary than SARS-CoV-2 or the flu virus, these different segments of the genome are also encapsulated in different capsids, so they propagate independently. And these viruses need to, like these different parts of the viruses need to co-infect together in order for the species to not get extincted, right? So this would be like a permanent association between the different particles, because otherwise the infection cannot proceed. So I'm going to put an example of this. So here we have two examples in figure 1. We have different leaves, as you see here, and we have, in this case, is bipartite viruses, so it's a virus that needs for the infection of two particles, in this case A and B, in order to proceed the infection. So here you have the leaf. When you only put virus A or the part A, nothing happens, the leaf is okay. Then when you have the two parts, then there is an infection. And then this beta is a satellite. So sometimes the satellite can appear, and if it appears, it seems that it enhances the infection of the bipartite species. Again, here we have another example that is the cucumber mosaic virus. And in this case, this is a virus that is composed by two parts. It's a bipartite species. So it is composed by two segments, independently encapsulated. And sometimes they can isolate these viruses with a satellite. And when this satellite is co-circulating with this virus, you see that the cucumber leaves are very damaged. So it's kind of like a satellite that exacerbates the infection of the virus. And in general, I just wanted to put another example, is that the case of thrombus viruses. So thrombus viruses are plant viruses, and they have a wide variety of satellites and associations. So you can have a thrombus virus that is associated with a satellite virus, with a small linear satellite mRNA, and also you can find bipartite or multipartite species. So in this case, a more permanent interaction between the entities of the co-infection. So when I was studying this in my PhD, what I was thinking is that, okay, there is some empirical observations that say that, like, we observe that many viral species and subviral entities can co-infect a plant, so they co-circulate together in the same plant. So how we can create a model about this, and why they have coexistence, why we can find these co-circulating entities, and we say that they can coexist, how this happens, right? So we propose this model. So in this case, we have a susceptible host that is a plant, H, that is in blue. And this plant grows at a certain rate, G, and decays as with a certain decay rate, D. And then this plant can be either infected by virus X and virus Y. So the virus X is in green and the virus Y is in yellow. And then these two plants that are infected by virus X and virus Y, they have an increased death rate because of the infection. So in this case, for the virus X, you will have an increased death by dx, which is the increased death of when the virus X infects the plant. And we can define two important measures here. So one measure is the invasion fitness of each of the viruses. That depends on the rate of infection, that is P, and the decay of the plant that is infected. And then we have another measure here, that is the host turnover, which is basically the time that you need to wait until all the host population is renovated. Okay, so there is a premise that we have here. So if we learn something from the models that we explained before, is that there is going to happen a competitive exclusion principle here, because we have two species, in this case, two viruses that are feeding on the same source, which is the same host. So we are kind of having this premise that they cannot stably coexist. So either one or the other would be will be able to invade, but there is not going to be, we are not going to observe coexistence. Okay, so let me first, before going to that, the results, let me first talk about the epidemic thresholds. This is something that if you haven't heard about that because of COVID, you should. So the epidemic threshold, or the reproductive number, we can define it, and it's related to the fitness of, or the invasion fitness of the virus. But what this reproductive number means is like how many hosts one infected host can produce. So it can have, it has to be larger than one, because it means that if it is one, it means that the host that is infected only infects one other host that is susceptible, and then let's say that the infection doesn't produce, or they don't evolve. So if it is larger than one, so in this case, for example, in the case of COVID, was around 2 or 3, depending on the strain. So then you have like an invasion, right, of the virus in the population. So we know that the reproduction numbers, or the epidemic thresholds, that are the same, it's just a synonym, of the virus X and the virus Y have to be larger than 1. But then we have something here. Here I plot the reproductive number of the virus X, and here is the reproductive number of the virus Y. And we see that at some point, if we change this parameter in the model, we see that beyond this point, the virus X, the virus Y have a higher a higher reproductive number than the other one. So let's see what happens with the results. So this is the parameter space, and the parameter space is depending on the growth of the host and the increased decay rate of the virus X. So these are two parameters that I just selected, but I could select any other parameters. It's just to see what are the solutions, or what are the solutions of the model depending on changing these two parameters, right? So first, we look at this region over here, the blue region. And in this blue region, both epidemic thresholds are from the virus X and the virus Y are less than 1. So then we will have a virus-free state. So host, like neither of the virus can infect the host. So the host is free of viruses. And then we have this limit over here, this limit that is in the dark, in black dash line, which corresponds to the limit or the threshold in which the reproductive number of X and Y is equal to 1. And then regions here. One region is where the virus X wins, and this only happens if the reproductive number of the virus X is larger than the reproductive number of the virus Y. So we would be in this situation here, right? In this part over here. And then the other way around. We have like the reproductive number of the virus Y is larger than the reproductive number of the virus X. So then the virus Y wins. So we were in this region over here, right? Okay. And then we have this line over here, this red dashed line. And in this red dashed line, it corresponds to this situation over here, which is when both epidemic thresholds of the two viruses are equal. And here we have coexistence, right? So if we look to the bifurcation diagram in which we have the different solutions, so in solid lines are the solutions, where the solutions are stable, and in dashed lines are when the solutions are unstable. So we can see that this line over here is this line over here. So we have like a coexistence solution, but this coexistence solution is just like relatively stable. Like if you introduce any kind of perturbation to the system, you are going to go either to this state or this state. So now let's complicate the system a little bit. So I'm going to add a satellite. So this satellite will depend on one of the viruses, in this case, the virus Y. So if we look to this diagram, we again have independent hosts or susceptible hosts that can be either infected by the virus X and the virus Y, the same way that we had in the previous model. Then we have another state here that we are going to call hosts that are infected by the virus Y and the satellite. So it's like the tandem. And because the satellite cannot infect by their own, it always depends on the virus, the helper virus. You are not going to have free satellite state, right? So let's define the invasion fitness here. So the invasion fitness are, again, the rates of infection divided by the decay rates of either of the viruses. And then we can define also another parameter here, which is the probability of infection of the tandem, like the satellite and the virus both together. Like they can also be transmitted in tandem. So if I have a plant that is infected by the satellite and the virus, the helper virus, in this case is the virus Y, if I am in close contact with another plant that is susceptible, there is a probability or there is a rate in which the two entities, the virus and the satellite, infect together. So we are going to call this PC. And we have another option that is that plants that are infected by the virus Y, they can get infected by the satellite because they are already infected by the virus Y. You can add the satellite or a satellite that is co-infected and then have a plant that is infected by both. So because you have here dependent and independent. Sorry, 1 second. Someone is calling. Sorry, I am in if you come just in 1 hour. Sorry for the introduction, the interruption. Again, what I'm saying is that PSY is the rate of the co-infection, like the satellite and the helper virus co-infecting together. And then we can have another option in which plants that are only infected by the virus Y, they can get infected by a satellite and then create these S plants that are infected by both the satellite and the virus. So what we are going to look is on how the equilibria that we observed before are going to be affected by the association between with the satellite, right? So what happens if the satellite doesn't have any effect? So we call this a commensal satellite. In this case, we have that the association or the fitness of the association of the virus and the satellite is a higher fitness than the original virus alone. So then we will have a mutualistic relationship between the virus and the satellite. And in the other case, we have a parasitic interaction if the fitness of the virus and the satellite together is smaller than the fitness of the original virus alone, which means that when the satellite is co-circulating, it's kind of like it acts as a parasite. So it's decreasing my fitness, right? The invasion fitness of the virus. So let's see the different scenarios that we will have. So again, we have our parameter space with the same parameters. So we have the growth of the hosts, of the susceptible hosts, and the increased decay rates of the virus X. And again, we have the same regions that we saw before. So we have the blue region, that is when the epidemic thresholds of all the viruses are smaller than 1. And then this line corresponds to when the epidemic thresholds are equal to 1. And yeah, so here we have the free virus, the region of when the Y virus wins, and the region when the X virus wins. So we just set the same solutions that the other model, the previous model without the satellite. And then we have this orange region, and this orange region happens when this is happening. So the fitness of the virus and the fitness of the combination of the virus and the satellite are equal. So we have just these cases, right? So again, this line over here is the new line that appears, and this line corresponds to all the epidemic thresholds are larger than 1, but we have this condition of coexistence in which the epidemic thresholds of X, Y, and the virus Y and the satellite are identical. So again, we have stable coexistence, but again, any perturbation in the environment will destroy this coexistence, right? So this is the bifurcation diagram. It looks very similar to the other diagram that we saw. But let's go to more interesting solutions. So in this case, we are going to consider a satellite that has a higher fitness, like the combination of when the satellite is present, the virus Y has a higher fitness than what it has alone. So we say that this satellite is a mutualistic satellite. And what we see is that a region that was previously only occupied by the virus X now has this region of stability. And this region of stability means that depending on the initial conditions of the system, you will end up having only X or the virus X and the satellite. So let's say that the mutualistic satellite can prevent the extinction of the helper virus in this region, particular region there. And the conditions for stability are, again, what I said, like the epidemic threshold has to be larger than 1, obviously. But in this case, the epidemic threshold of the combination of the virus and the satellite has to be larger than the competitor. So yeah, so in the limits of this region of stability, obviously, is going to be when the epidemic thresholds of the combination of virus and satellite is equal to the competitor virus, the virus X. And this is the bifurcation diagram. So in the bifurcation diagram, you can see that there is two solutions that are coexist, that are possible. And finally, we're going to talk about what happens with a parasitic satellite. So in this case, when there is a co-circulating satellite, this is going to decrease the fitness invasion of the original virus or the helper virus. So that's why we have this condition over here. And then again, what I'm finding in this parameter space. So what I'm finding is that there is a new region here of coexistence. And this is not just arbitrary coexistence or point coexistence. This is a real region of coexistence. And when this region of coexistence happens, so these regions of coexistence happen with a wide variety of parameters. The only thing that it has to, like, the only condition is that all the epidemic thresholds obviously are larger than 1, but the combination of the virus and satellite has the lowest epidemic threshold. So if this happens, so basically, if the satellite is a parasite, the same way that we had before in the other examples, if the satellite is a parasite, we observe coexistence. And this is on all scales. So every time that you have two species that are feeding from the same source, if you have a parasite that controls the species that is kind of like the better fitted, you are going. So let's say that, yeah, exactly. So basically what I'm trying to say is that this phenomenon that we are observing here is applied to all the ecological scales in which you have one niche, and from this niche, 2 or more species are feeding or are coexisting, and they can only coexist if there is parasites that control the species that grow better, that have a higher fitness. So the conclusions that I have from this is that in this model that we just presented, it's kind of different from the predator-prey model of Lotka-Volterra that we observed, because there is not oscillating coexistence. And also we have like a free epidemic state. So we have free independent hosts. Then another conclusion of this model would be that the virus and satellite associations can promote long-term coexistence. So this is what we just observed, that if we have a satellite that is a parasite, we will observe in the long-term coexistence of competing species of viruses. And this obviously depends on the phenotypic modification. So the phenotypes that we observe from these parasites are going to really promote or not promote the coexistence, right? So I just want to acknowledge my previous supervisors, Susana Manrubia and Jacobo Aguirre, that was the postdoc that was helping me with this model. Now I am in the weights group, as Flavia said. We have these findings, like arrow finding and the CDC finding. So I don't know if I was very fast, sorry about that. Yeah, so now we can talk about questions. You can ask questions if you have any.
Okay, thank you so much. Are there questions, guys? Where is the mic? Pass the mic.
Hello, Adriana. It's Adenilson. I hope you're doing fine. Thank you very much for the presentation. I have a very basic question. I think in the first differential system, we had these outflow terms. What do they mean?
Sorry, in which system?
I think it's the first one.
The first one, you mean of the second?
SIV model.
Okay. Wait a second. I'm going to go there. I don't know how to. I'm sharing my screen now, right? Okay.
Wait, yeah, now we see.
Here, right?
Yes.
You can see the slide?
Yes. It's that.
Okay, so the SIV model is a compartmental model that has the similar dynamics of the Lotka-Volcker, so the predator-prey. So here we have three compartments: susceptible cells, infected cells, and then free viruses, right? So here is what we have: susceptible. The growth here is a logistic growth. So we have a exponential reproduction, right, of the bacteria, and then they will grow until until they reach the carrying capacity, right? A maximum growth. So is when the limiting sources, you cannot have more growth than what the environment sources gives you. Then you will have an absorption rate. So this absorption rate means like the infection rate of the virus. So is the rate at which viruses infect or get inside of the cell. And then you have here the outflow, that means the decay of cells. So these cells eventually die, right? Then the infected cells or the infected bacteria would be whatever I infected, right? Whatever the free viruses have infected. And then you have here a parameter of lysis. That means that you deplete these, like you kill the bacteria. But the bacteria can also die by normal conditions. You will still have the outflow or the decay rate. And then for the virus part, we have this term here, which is the burst. So the burst will depend on the rate of the lysis, so how fast the viruses, when the viruses are infecting the cell, how fast they kill the cell. And then I have here this parameter B that we call burst, that is how many times the virus replicates inside of the cell, right? So how many new viral particles I generate from one infected or from one infection, from one virus that enters the cell. Here I have another absorption. So this means that there are some viruses that absorb the cells but don't lead into an infection. And then also I have a decay of the virus because, they can also decay normally.
Okay. Thank you very much. I would like also to ask about the machine learning models you're using.
Yeah, so that's why I didn't want to say anything because the time to put everything. But basically, in the machine learning algorithm, if I go back here, I don't have anything. Like I don't even have the equations or anything. Sorry about that. So what I'm doing is that from the experiments, they analyze each of these dots, right, correspond to an infection, an infection between one genotype of E. coli and one genotype of PAI, right? So they sequence all these genotypes so they know which mutations, or they know the evolution phylogenies of both, right, E. coli and lambda phage. So from them, what we can forecast is what is the mutations that are important in order to reconstruct the infection matrix. So in this model is an additive model. So it means that I am going to add mutations that belong to E. coli to the bacteria, and mutations that add to the fate. Yeah. So what I'm saying is that both mutations are important and they add. It's not like they have an additive effect. This is the model that I am construction here.
Okay.
I don't know if
thank
that
you.
on your questions.
Okay, and just one more question about the I think the last model you are using. Yeah, the last model of differential equations.
Yeah, sorry. Here.
Okay, that one. There you're modeling the number of susceptible hosts infected with X, with I, and with the I plus the satality.
Yeah.
What I'm going to say, I think is not a good suggestion, but I'm just curious about it. Did you think about modeling also the number of virus and the number of satellites, or would be like too much parameters?
You mean like 3 viruses and 3 satellites?
Yes.
I mean, yes, I could have done that. I could have done that. But we can say that when you use when you use like this kind of model in which you implicitly have the infection, I believe that, I mean, the dynamics become much easier, as I said. That's the reason why I could do analytical work. If you increase this number of equations, now we have 4 equations and we have kind of like several parameters already. So if you increase the number of equations, I didn't try to do it, to be honest, and I don't think what would be the purpose of it. Probably you will have similar equilibria, but then you will have some kind of different parts because, for example, I don't believe that you will have a pre-epidemic state. So what I'm trying to say is that, yes, I could have done it. I didn't do it because this is more simple. If I had done it, like if you want to include this, like if you want to include the pre-satellites and free viruses, then you will have other type of dynamics that would be also oscillating dynamics. So yeah, I mean, the reason why I selected this is more simple. If you complicate more the dynamics, you will have different outcomes, I will say. Like some of them will be similar, some of them will be completely different. And I believe there is some kind of some papers that have modeled that already. Like I believe Joshua and another student of Joshua have done something in the past similar.
Okay, thank you very much. I think in general, simpler is better.
Hi, thank you for your presentation, Adriana. I would like to ask you to comment a little bit further about the virus microbe interaction network plot. It wasn't really clear for me when you talk about the modular and nestedness of the interactions. I think it's the plot right before the slide that you were showing at the Nielsen. This one. Yes.
Okay. So this is multi-scale interaction network in which like they took samples from the sea, from different bacteria. They were almost all Vibrio bacteria, and then fakes that infect those bacteria. Okay. So what they observed is that instead of seeing a nested infection network that like this one, right? They observe nested infection networks in here, but then on the large scale, when you just zoom out from the different families, so here, this is one family of Vibrio, this is another family of Vibrio, this is another family of Vibrio, and so on, so forth. What they observe is that the different taxonomical levels have different infection matrices. So if I zoom in this module, the blue one, I will see a nested network. But if I zoom out and I go to different taxonomical levels, which means I go to different families of bacteria, then they separate by themselves. So it's not very probable, like here you have these little dots, for example, you can see here. So this phage over here, they can infect this particular bacteria, but it's very rare that they are going to infect bacteria that are very far away in phylogeny. So they are very different taxonomically. So that's why you have this kind of diagonal. That means that phages are going to infect bacteria. So similar phages are going to infect similar bacteria. And that's the reason of the diagonal. I don't know if this is clearer.
Okay, it is very clear. Thank you.
Okay.
There's another question. Oh, you have the mic.
Hi, Adriana. Thank you for the talk. I want to ask you about if it's possible to consider the action of antibodies or a medicine in the model, and how is it possible to incorporate it, like adding other compartment or other parameter?
Yeah, so that's a very good question, because as you know, there is this new alternative antibiotics that are with phage. So like, you know that there is a crisis of antibiotics and we need alternative methods to treat persistent bacterial infections, but when the antibiotics fail, so many people, or at least they are starting to use phage therapy. And in these cases, you have to consider what happens with the immune system. So yes, I believe, so Rogelio is one of the students here, and he has done work specifically on that. So you will have a bacteria, and then you will have phage that infect this bacteria, but this bacteria can become resistant. And then you have like the effect of the antibiotics and the effect of the immune system. And depending on how the immune system works, you will have a clearance of the infection of bacteria. So let's say that the person survives of the infection, and in other cases in which it dies out. So, I mean, these kind of models are already existing, and there is like some publications. So you go to the website of Joshua, or website of the group, you will see that there is a part that is about phage therapy, and you will have these kind of models too, in which you consider the immune system.
Thank you, Adriana.
The question?
Yeah, I do.
João, yeah.
Hi, Adriana. I have a couple questions. I don't know much about satellites. Honestly, I don't think I have heard of them before today. But I had a couple questions. So, are they like host specific? Are they specialized in a single kind of virus, or could the same satellite infect X and Y, like in your model, for example?
So far, like, this is kind of a new thing, right? It's like we are now on what would be like part of my thesis, that was kind of like what was new in the world at like 2 years ago, right? Or 3 years ago. Now the things have changed so fast and rapidly. But normally these satellites are specific of the virus. So they need a specific helper virus to survive. Like they cannot they are specific to the virus, yes.
But that comes back to like the question of Isabel. Like similar viruses infect similar bacteria, so could like a satellite infect like similar viruses, for example?
To be honest, I have no clue. I just talk on the empirical observations. So the empirical observations, you didn't, there is no isolation of satellites that infect multiple or can infect with multiple different species of viruses, but I don't know if that's true or not, to be honest. Like it could be, but it haven't been isolated.
Okay, and final question, what's the scale that you were working with when you're talking about the plants? Are you talking about different individuals or different cells of the plants? I didn't quite get that about the study.
Yeah, no, in the study, I consider the whole plant. So it would be a field of plants, each host, like here, sorry. So each H is a different plant. So H will be plants that are susceptible plants. X are plants that are infected by the virus X, and Y plants are plants infected by the virus Y.
Thanks, Adriana.
Hi, Adriana. Thanks for the presentation. I was also very curious about satellite virus. And my question is, do we actually observe in nature this kind of viral competition involving satellites? Okay.
Yeah, this is what I don't know who was the person that was talking before, but the partition, you mean like the partition in taxonomical levels? Sorry, I didn't understand very well.
Sorry, the viral competition.
Yeah.
Do we observe this kind of competition involving virus and a satellite? Could you give us an example, maybe?
Yeah. So the examples were over. So here are just observations that are in nature. So again, what I was trying to say is that I don't know if you can see my pointer. I don't see it, so it's kind of weird. Wait a second.
We don't see your pointer.
Yeah, me neither. I don't know how. I'm going to stop sharing 1 second to see what's going on. I see what is happening. Okay, sorry. Okay, so now you can see some kind of pointer, right? Okay, so here what we have is different options in which you have, let's look at this, just at the second example. So here we have a virus that is cucumber mosaic virus. So this virus infects cucumber plants. When there is a satellite co-circulating with the cucumber mosaic virus, it changes the etiology of the infection. So the infection is more severe. So this plant, when you look at this plant, in the cells of this plant, you will isolate both the cucumber mosaic virus and the satellite. Okay, so yes, there is evidence that in one plant, it's not only one virus, it's many viruses. Actually, this is something that is very interesting in plants. Like normally in other hosts, like for example in bacteria, it's kind of rare that you have co-infection of different families of viruses because of what we talked before. That is kind of rare that a specific virus strain or a specific phage can infect very distant bacteria in evolutionary terms, or very different families of bacteria. But in the case of plants, many viruses can infect the same plant in different ways. So when you look at plants, especially in crops, in plants that humans are interested in, you see that there is a lot of plagues, a lot of viruses that are infecting them. And you can isolate many viruses in only one plant, but many viruses of many different families. So the coexistence or the ecological coexistence in plants is super high compared to other situations, like, for example, when the host is a bacteria. Okay? So that's the reason that I put here these examples on plants, because they are the virus of plants are very promiscuous, let's say. I don't know if that was your...
Yes, thanks. I before I didn't get the example. Now I understand. Thank you.
Any other question, guys? I have. Adriana, I'm here, just below you. You don't see me, but I'm here in the front of the class. It's you on the top and me down here. So when you showed the bipartite network of hosts and the viruses, I realized that network is actually something that maybe is not coexisting in time. Is it true?
Are you talking about this?
No, the one that you showed before, like very graphic before.
Before?
Yeah, like the bipart. Yeah, this one and
this or
yes,
this?
the nested one.
This one?
The slide 16.
16. Okay.
Yeah.
Here. Okay, so here what I was proposing is a type of coevolution network in which you have an original bacteria, an original virus, phage. So this phage is able to infect this bacteria. So when it infects this bacteria die. So then evolution is pushing this bacteria to be resistant of this phage, right? So through mutation, this bacteria will evolve resistance, so it's not longer infected by the original virus. But then over time, this virus is also mutating, right? And eventually it will mutate into a strain of viruses that is able to infect this resistant bacteria. And again, in time, this bacteria again will gain more resistance. So this bacteria is resistant to the original phage and the original and the second subpopulation of phage. But again, this is like an arms race, right? And these arrows over here, what I am trying to say is that evolved viruses conserve some kind of memory of previous bacteria, so bacteria that were in the past. And the reason is because they are still co-circulating. So imagine you have an experiment and you have original bacteria that are evolving over time, and original phage that is also evolving over time, like to be able to infect resistant bacteria that are emerging. But the problem in together. So this is like not real evolution. It's real evolution, but what I'm saying is that all are coexisting together. So all are in the same environment, like all bacteria. There is a problem, so phages can also need to be infecting other past emergence of a nested network in which new phage, like new phage over here, this is the old phage. So this old phage only infects the original bacteria. So this is kind of switch a little bit. So this original phage only infects this original bacteria. The original bacteria is infected by all the phage, ones. And this is the most
Oops. I think you are down now. Yeah. So we'll just wait 5 minutes just to see if she's
Sorry, I
back.
think that I lost my connection.
Yes, and we don't know where you are.
Hello?
Hello? Adriana? Hi, Adriana. We don't listen to you anymore for some reason, and we don't see your screen as well. Guys, we are waiting 5 minutes just to check if she'll be back. I think she's really down now. And we are waiting this 5 minutes, and after that I have a quick message for you. So
Sorry, I just was lost, but I came back.
hi, Adriana. Yeah, now we see you. Perfect.
Sorry about that.
No, don't worry. You were talking
Yeah,
about
I
like
don't know
the bipartite network, and
yeah, I don't know if you heard things that I said or not.
I think the very end that we didn't listen anything because you were frozen. The message was cut. But I think I got the main message, and I have just one more question. It's just because, as everyone else may be here, I never heard about satellite. And I was curious about, so you showed the experiments that some people did with the plants. And I was curious if is it possible to do some experiment where you include the satellite inside the plant somehow without the other virus?
Yeah, so in that case, what I was saying, because satellites are dependent on one virus, the helper virus, they cannot reproduce it on their own. And the reason is because they lack from the necessary proteins or machinery or genetic material to accomplish their own infection process. So they need from another virus that supplements these failures that they have or these lacks.
Okay, it's for both to enter and for the reproduction, is it?
Yeah, either to be transmitted, so to like create a viral particle, or either to replicate. They need it.
Okay, great. And about the teaser part, because of course you gave us a teaser, so I realized that in that how do we call it, that matrix, the red part was in a very specific part of the time, and this is not in the end, in the very end, and this is not in the very beginning, it's in the middle of the time of the evolution. Do you have any clue of what is happening there?
Yeah, basically what is happening is that the more that you have your virus incubating with the host, the bacteria, they become better at infecting. They become better and better. Like this is just like part of the evolution process.
Okay,
sorry.
you're back.
I can put again my screen.
No,
but
no, don't worry. I think I understand that. Oh, that's really cool. I want to hear more about this in the future.
Yeah, soon the paper will be coming out. So
Cool. Any other question here? No? So let me help to thank you, Adriana, again.
Thank you.
Thank you very much, Adriana. Ciao, ciao.
Yeah. Thank you, and nice to see you all. Good luck.
Thank you.
Bye.
Bye. I have a message, guys. I just need to know how to finish this. Oh, I need to stop.